Welcome, everyone. I'm here with Dr. Yumei Huang of CellMosaic Inc., and we're delighted to have you here today with us to talk about how CellMosaic is helping make cancer drugs safer. So I'm just going to start with a couple of brief housekeeping items. At the end, we're going to have a Q&A. And in order to ask your questions, don't use the chat button. There's a Q&A button down below. And so please, at any time, you can start asking your questions and we'll get around to answering them later on at the end. I am so excited to be here presenting with CellMosaic today. I think this is such an outstanding company. Right now, they're open for an investment opportunity on Start Engine, but the campaign will not last forever. It's actually not going to be open for too much very longer. So I'm glad you're here today to learn more about it. Dr. Yumei Huang has a very impressive background in chemistry and biology with over 25 years of experience. She founded CellMosaic back in 2008. Has a PhD from Case Western Reserve University, did some pretty impressive postdoctoral work at MIT with a Nobel laureate. All of this experience and expertise has helped her develop over 220 proprietary processes for developing bioconjugates. It's kind of a technical thing in the medical space, really with advanced medicines. Very interesting. It's helping drugs stay, she'll get into the technical part of it later, but suffice it to say, they've developed over 200 special methods for helping drugs work better and secured 12 patents around it. So now I'm going to turn it over to Yumei to talk about the business model, what they've been up to, really why they got started in all this. And yep, I'm going to drop the link to Start Engine if you want to learn more over on the investment offering page. Again, Yumei, I'm going to turn it over to you.
Sure, let me just share my screen first. Okay, thank you, Abby. I'm so happy all you guys can join here. And so today, CellMosaic is a revenue generating company. It's located in the Great Boston area. So today, I will spend some time talk about the problems for the current cancer medicine treatment and our technology and the solution. And then towards to the end, I will talk a little bit about the marketing and our attractions and the business plan. Before diving in, since we have a regulatory CF offerings through Start Engine. So this is the standard disclaimer I have to show here. So please read carefully. So the investment is risky and including the possible loss of your entire investment. So what is the problem of current cancer medicine treatment? So in 2015, so my mom was diagnosed with a nasopharyngeal cancer, which is a cancer located basically below the nose and up to the throat. And the cancer is kind of spread to her bone. So I was sitting in a room with the best oncologists and the radiologists from Massachusetts General Hospital. I was so surprised to find out, despite all the advancement and all the research going into the cancer research, the only option she got is chemotherapy. And it's not also the normal chemotherapy, it's the cisplatin, which shows here. And the cisplatin is really toxic compound, which because most of these chemotherapy drugs, they are nonspecific. They affect all fast-growing cells. In terms of for the cisplatin itself, they basically target all the genes, like basically the oligo, they are the oligo-accumulating reagent. So it's really, they have a lot of harmful side effects. And my mom, 10 years ago, she had a stroke. So she has half of her basically body is kind of paralyzed. And she also have a diabetic. So doctors, and we all know that if we're going to give her the cisplatin, she will be immediately end up in the bed. There is no way she can survive the whole cancer treatment. So eventually we settled down with a less toxic compound called carboplatin, which is 1/10 toxic, like effective basically than the cisplatin. But the problem is we don't know is that going to work, is going to save her life. Fortunately, after 3 months of chemo plus the radiotherapy, and she was declared cancer-free, and then the cancer kind of all gone. But within 1 year, after 1 year, so she died suddenly, just four, and then she died. So we thought probably through the stroke or heart attack. But in back of my mind, I was always wondering, is really because of that, or is just because of the cancer treatment, the chemo caused all the side effect actually affect how she was in her blood and all this stuff. So that's why it caused the stroke. So anyway, so take back home messages, chemotherapy is a non-specific treatment. And then on the other hand, the chemotherapy, most of the drugs, they don't mix well with the bloodstream. So when they transport it, so most of the chemo, so you have to put this, like when my mom get the cancer treatment, they always have a little pore, so you can basically put the drug go directly through bloodstream. So the chemotherapy drugs don't mix up with bloodstream, so they will have instability when during the transport and increase the side effect. So in 1986, the first target therapy called immunotherapy using like only antibody was approved by the FDA. This kind of opened a new ways of treatment and is less toxic compared to chemotherapy. And some of you might watch the movie called Living Proof. So the movie talked about Dr. Dennis from UCLA and his journey to discover this amazing drug called Herceptin. So what Herceptin does is, so when you look in the cancer cell, right, the cancer cell in the surface, they have all this receptor, which basically, so if you have HER2 positive, so it's what do you call it, like express that receptor, and Herceptin can bind that and then cause an immune response and then kind of like recruiting other immune, the one to kill the cells. But think about it, because the mechanism is always by, you bound, and you can also get released. If there is one get released and one did not get escaped, that would be devastating, because the cells might transfer to somewhere else, cause the metastatics. So antibody therapy, if they used alone, is often ineffective. They typically requires combination with chemotherapy, so the ultimately not curable. But if not curable, as I said, that it can lead to drug resistance, meaning that the cancer cells will spread to other part of body, so making further treatment more challenging. So antibody drug conjugate is really combine the benefit of the chemotherapy and the immunotherapy. So if you look at the right side, these pictures, so antibody drug conjugate contains 3 components, the antibody, linker, and drug. If you consider the whole thing as a smart package in a post-delivery system, the drug itself is the package, right, that provide the treatment you need. And then the antibody is the address label. They can guide the drugs to the cancer cell. And the linker between them is the package tape. So controls how the drug will be delivered. You want the package to be secure right before delivered to the right address. So the first drug called Myotag, this is from Pfizer, was approved by FDA in 2000. And in Myotag, actually only half of the antibody has the drug inside. This is the first generation drug. So Pfizer actually acquired the Wise pharmaceutical to get that drug. But that time, because the ADC kind of process is still not very developed, so until Seattle Genetics, which is the company who got the second ADC get approved in 2011, and the Immunogen, who helped the Genentech to get the SAT ADCs approved. These 2 companies is really the, I think, early pioneers in ADC technology. So they are the ones, Seattle Genetic and Immunogen, both of them, they're the one who established a lot of the linkers and then the drug toxin they use for the ADC today. And both company was sold in 2023. Seattle Genetics sold to Pfizer for over $30 billion, and Immunogen sold to the Abbvie for around $10 billion. All of this drug has annual sales between 1 to 2 billion in 2023. So the challenging is because all these current ADCs in clinical trial and commercial trial is all kind of using the technology developing by the Seattle Genetics. So they use a linker system which is not supportive of effective ADC delivery. So the linker itself is not really water soluble. So if you look at here, all the drugs, they're like oil, they're kind of sticking together, right? And then that's caused all the ADCs come together. So now you get many, if they have a lot of ADCs stuck together, they will eventually precipitate out, leading to the side effect, build up the bloodstream. And also because of the tape, which we just talked, the linker, the tape is not helping with the solubility. So to compensate that, the pharmaceutical companies are trying to lower the drug loading, basically just put less drugs into an antibody. And then to compensate the low loading, they use highly toxic compound, which called a toxin. And these toxins is 1,000 times more potent than the normal chemotherapy. So imagine that if one of the ADCs with these toxins is kind of get lost somewhere, did not find the right address, and then the result is devastating. So the impact on patient outcome is really very serious. So today's ADCs are primarily approved as a last resort treatment when all other therapies have failed. They're mainly used for metastatic cancer when the cancer is already spread to other parts of the body, so which significantly limits their full potential. So at CellMosaic, we have been trying to deliver a linker system which can solve this problem. So we don't want just one linker as Seattle Genetics and Immunogen did. And we want actually a linker system which can be tunable, can, versatile, and we can use it for customize it for all kinds of different drugs depending on the property of the other drugs. So in 2011, I was sitting in the Indian restaurant at Warsaw, just outside of Boston, with Andrew Mark, who used to be the chief patent officer from Vertex Pharmaceuticals. So I was asking his help to draft the claims for that. When I told her that we're going to use this sugar alcohol, which is, right here I give you an example, is mannitol, is like sweetener. We've used that as a monomer, and then we assemble all kinds of structures, linker structures. He was like, very surprised. There is no other people ever think about it. I said, no. So that's why, we are so lucky because we have already, went through all the system and look into that. We know that there is no prior art, not too much prior art, the one. So our patent is easily get allowed. So far we have like 11 issued and allowed patent worldwide, cover the broad composition method of AQT platform. And in 2014, we decided to apply one of the most competitive grants by DoD. So it's called a Department of Defense Breast Cancer Research Program Breakthrough Award. So they have 3 levels. Each year, like not more than one nonprofit companies were able to get the grant. So we're fortunate enough. So in 2014, that year, we're the only nonprofit to obtain to get the recommended for the funding for the level 1. And as I said that, we are also the first and only company in the sugar alcohol IP space. So what AqueaTether does to the ADC is very obvious, right? When you have the linkers with all kinds of hydroxy group there, so it is really built up, we call a hydrogen bond network. It's kind of built like a wall to shade away these drugs, so they don't stack to each other, they don't stuck to each other. the end result is you get this AqueaTether. Each one of the ADC is happy. They can stay stable on the solution, so there is no clump formation. On the other hand, most in the blood systems, you have a lot of kind of enzyme like protease, which can degrade of this antibody. So what AqueaTether does, they can this network which build up this will be also stop the enzyme kind of to access on that, and then you can basically prevent these biomolecules from enzymatic breakdown. So the other good features for this AqueaTether is they can enable integration of multiple drug types in a single ADC. So in that cases, you can use a combination of the chemotherapy drugs to within a single ADC. If we can use, since the ADC's AqueaTether allows the high loading, we can also allow the use of less toxicity drugs, which is the key to reduce reliance on potency toxins. The outcome for patient care is outstanding. So basically now you have reduced the harmful side effect. You have increased efficacy of drug treatment. So potentially we can transform the ADC from last resort treatment into standard care. That would be helping tremendously prevent the metastasis. So CellMosaic founded in 2008 is with an urgent mission. We want to make the cancer treatments more effective and less toxic. So beyond the ADC, we use bioconjugation as a general tool to attach, drugs to a target molecule, create a bioconjugate we call a smart package that can find and treat specific cells in the body where living healthy cells are unharmed. And then this smart package will enable precision medicine. They can reduce side effects. They can make existing drugs more effective. They can also open new possibilities for treatment. Over the years, we have developed over 220 proprietary processes that helping us to create these precise delivery smart packages. We make this expertise accessible through PackKit technology platform. So the PackKit is basically the commercial kit we sell on our website, provides researchers with an efficient, cost-effective way to create smart package in their own labs. So we sell with customers ranging from university labs and biotech startups and to global pharmaceutical companies. We also work directly with customers to collaborate on improving and developing their drugs. So additionally, we also are developing other kind of platforms for drug development and safe testing. For example, the OxLink and XLink, which was funded by the NIH Small Business Innovation Grant. This technology helps scientists understand how proteins interact with each other, and we enable pharmaceutical companies to identify better drug targets and design more effective medicines. Neo platform, on the other hand, solves a unique challenge. They're tracking just tiny amounts of drug in the body. So when you take medicines, your body breaks it down into small, tiny pieces called metabolites. So Neo makes these pieces easier to detect and measure, helping researchers understand how the drug moves through the body and how they might cause side effects. This is particularly important for developing safer medicines with few unwanted effects. So all these additional tools demonstrated CellMosaic's broad expertise in creating technologies that make drug development more efficient and effective. So what started as a mission to improve cancer treatment has grown into something much bigger. So we believe our technology could help deliver many types of medicines more effectively, from vaccines, for opium addiction to treatment for diabetes and heart disease. And so not just the antibody drug conjugate, you can use a peptide as targeting molecules, you can use oligo as targeting molecules, you can do peptide drug conjugate, you can do peptide oligo conjugate. You can also use antibody oligo conjugate, this for all these gene therapies. And if you look at the example here, this is Adequine and the Neurolaster. So these two is commercial drugs, is all $1 billion drugs. So this is so they use a molecule called PEG to modify it to make them stable into the body, but the PEC has this they generate the immunology, and then they are really not recommended in this case. So our AqueaTether can do the same thing and then doing better job on that. So there's a lot of opportunities for us here in terms of all kinds of smart package. For the market side, so the ADC market estimated to 40 billion by 2029. On the subset side, ADC deal values alone increased 400% from 2017 to 2022, reaching 60.6 billion in 2022. The contract manufacturing for ADC represent around 9.7 billion opportunities. So the total addressable the drug delivery is surpassed like 200 billion across all applications. So over the years, we have built relationships with 500 plus global customers across 34 countries, including 48 distributors in this 200 billion market. And our customers, including from biotech, pharmaceutical companies, educational, and then research labs, and also diagnostic clinical labs. So it's a very broad customer base. And also most of these customers, they're using all kinds of service and products from us. So we have like 159 organizations used our ADC reagents and kits. We have 16 organizations relied on our ADC customer synthesis and contract manufacturing. We have 38 organizations used our service to create other smart package for drug delivery. And we have 71 organizations use our service for non-drug related bioconjugates. So our technology platform serves as the foundation for developing a new generation of precision treatments. But we don't want to just, developing drugs ourselves. Our business strategy focuses on, so it's low risk, high potential approaches. So by providing essential tools for others to create medicine without incurring the cost of drug development ourselves, we empower scientists by offering essential tools to explore new ideas and grow alongside them as their projects advance. This business model is scalable. We started with basic research tools and then expand into custom manufacturing development service. We generate revenues at every stage of the drug development process. So if you're looking at our revenue streams, we sell research tools and kits currently used by 388 organizations, and the packaged ADCs are used by 159 plus organizations to generate ADCs. Our customer service has been served for 104 organizations. So far we have 6.9 million plus in revenue generated to date. That's validating like our business model and technology platform. In terms of the business growth strategy, we focus on our current established customer base and grow from there. Since our company has really strong IP portfolio and our technology protected by 12 patents, and we have over 220 plus proprietary process. So we aim to build up partnership that designed to create a large license opportunities. Beyond the ADC, we're also trying to, go to applications across multiple therapeutic areas. So I can tell some success stories with leading institutions and commercial companies. So we have established early partnerships with 2 undisclosed organizations and 1 pending collaboration to develop AqueADC precision treatments. So this is a kind of early partnership, which for proof of concept studies for their drug asset. And we have already generated revenue by supplying the materials to them for the testing. All these partners begin as customers using our packaged ADC products before advance to the AQT platform. This really kind of validate our business model and technology. In the academic side, since 2019, we partner with Professor Catherine Fieser's lab from University of New Mexico. We collaborated on the NIH-funded vaccine development for opiate addiction. The work that has been published on the vaccine in 2024, basically early last year. And we have another one come up, maybe within a few months will come up. We also collaborate with globally. One of the notable collaboration is with Dr. Goldie's lab from German Cancer Research Center. We helping them basically design and then create oligodrug smart package for brain cancer treatment, and the data works very well. So that publication also will coming soon, and we also she also have some other drug asset which we're trying to help them out. In the commercial side, company, we have helped us IBX Bioscience since 2021 to support their manufacturing of complex ADCs for in vitro and in vivo testing. And for real foods, and we have worked with Dr. Daniel since 2017. That's the interesting projects which Dr. Daniel is continue working on, and he's trying to develop in the strip test for the food safety testing. So unlike a traditional biotech investment that bet on single drug candidates, we generate revenue throughout the drug development process. So currently we're raising fund is mainly for you to build various equity-linked safe drug assets for current and future ADC partnering and licensing. We also develop and manufacture equity ADCs for preclinical and clinical studies. Finally, we want to support the development and manufacturing, expand global sales of other equity bioconjugate research products. So overall, CellMosaic presents a timely investment opportunities for the investors. We believe our combination of approved technology, established revenue streams, and significant market opportunities positions us to potentially capture increased value in the expanding drug delivery market. So we invite you to join us to get on the journey to revolutionize the whole the medicine treatment. Thank you very much. So I can take question now.
Great. Yep. So I wanted to remind everybody that we're using the Q&A function here on Zoom. So go ahead and add your questions if you have them. Yumei, thank you so much for sharing with us today. It's really interesting to hear about how you kind of came into this, and really I'm impressed by the strength of the partnerships that you've built. I have a first question here. It's about technology differentiation. And that the question is, what makes your solution different than what else is out there?
Well, I would not say my solution is different from other. I would say that everybody is trying to find a solution for that. It's just nobody had a solution. So the solution is very clear. Everybody knows that in the ADC cases on the drug delivery, you will need this super hydrophilic linkers. So the people is spend years trying to deal with it. It's just nobody really has it. So there is one company who has one called Mesana, which has this type of technology, but they are using polymer. I mean, they're not kind of tunable. So that's the only other companies which kind of like equivalent in our sense with doing this like massive kind of drug linker problem.
Okay, so in terms of competitive differentiation, there's really only one other company out there they're similar but not similar.
Yeah, so Mesana is mainly using for like they're mainly like a polymer type. So they use normal the polymer, but the polymer itself is not very stable. And unlike us, we actually really making the distinct what do you call the molecules, the linkers, and the meanings of that.
Maybe you could kind of help me understand a little bit about the technology platform. So you've got multiple technology platforms, right? And then it could potentially apply to a huge variety of different medical applications. Am I understanding that right?
Yeah, that's correct. Because the bioconjugate, which we kind of say double it as a smart package, and they are really used across the whole life science, from the drug development to drug delivery and the diagnostic and also research samples. So it's a huge market because and then we want to helping like the different industries or different companies to actually just capture values on that. Although but we don't want to do the drug development ourselves, but we can kind of capture the each step when they need a proof of concept and then we supply the reagent tools for them to use, so they can do quickly. And then when they move on to the production, manufacturing, and then they can come to us, and then we can help them to produce. And then if they need to do more innovative one, and so we have our proprietary linkers, so they can license it out. They can make a new drug so much faster. They can get the patent, kind of drug to differentiate from the rest of the industry.
Are you saying that sort of your role is to help other companies make great medicines, and that the way that you've set up your business model kind of makes it easy for them to dip a toe in the water with their kits and do some testing, and then they can decide to, if it works, right, then you can move on to the licensing phase. Maybe you could kind of talk us through the regulatory pathway.
Yes. So if you think about it, I mean, like the traditional biotech business model is always bet on one drug, right? So I have this, I want to make the assay. If the drug dies, I dies, right? The company says. I mean, the cancer is such a big problem, and all the medicine. You need a lot of smart people to do it. You can't do it alone. I mean, I'm very good in the bioconjugation. I'm good on my linker technology. I can make the one, but I'm not good in antibody because like I said, the customer. But if you think about it, so you have to pull all these things together. If you want it faster, you want everybody on the same page. You want helping all these people. So you want to have people who has the antibody, who want to develop in the ADCs. You also want people who has the drug, they want to test their drug. So by that way, you can kind of put everybody together, and then all the smart peoples, and then all the companies who work on that, you can push them so much faster. Because most of company, like antibody company, they really want to test if the ADC works or not, but they don't know how to make an ADC, right? So, and then we give them tool, they can do it quick. So a lot of cases, they can get it done within like 1 day and 2 days, they will have molecules on hand, they can test it. So they don't need to spend tons of money trying to rely on some other CROs to do that. And then we also, the most important part, we have the proprietary technology, the AqueaTether, which we know is going to change the way how all the bioconjugates we're going to do that. In order to make it popular, I have to work with a lot of people, so I can get them to popularize it, and then so they can get on board, and then so we can get better medicines. So,
and you've really got
it's one people's effort. You want everybody's effort being
an incredible list of partnerships. I mean, over 500 global organizations. And maybe you could tell us a little bit about the model for your distributors.
So the distributors, so we have right now, I think 48 distributors across 17 countries. So that's usually is how we say that, it's usually kind of some customers. For example, pharmaceutical companies, when they buy it from us, a lot of cases they have a lot of regulatory kind of other things that they would rather go through, like VWR, Fisher, which they already established relationship. So they go through that. And then on the other hand, like in the countries like Israel, like South Korea and Taiwan, and they don't want to deal with all the logistics, right, for the ones, so they go to distributor. So the distributor, so we actually don't know how many actually companies behind the distributor who is working with them. So the 500 we're talking about is it's just also including the distributors. The actual number of customers which we sell, like how many people doing all this innovative, we don't have the number. Yeah.
I want to encourage everybody to add your questions here should they come up. It's down here in the Q&A section. We have a few more minutes, so you can still add your question if there's anything in particular. One thing we haven't talked about yet is your patent protection and proprietary, the sort of moat you have around your intellectual property. Can you kind of tell us a little bit about how you are thinking about protecting your specialty?
Right. So AqueaTether, which it's interesting. So we using, I talk a little bit in that part. We use this sugar alcohol molecules. We make all kinds of linkers. the good part, so we have done, a lot of research when we starting writing even application for like the patent application. It's pretty much, it's clean. It's sled clean. Nobody has ever done it. Nobody has used it. So basically there is we have total freedom of operation. And it's very easy for us to actually get the patent. But the only hard part is it's very difficult to make the compound to actually get. So it took us another 10 years to build up the process to make the one. So we have two things to protect. First is the patent, which protect all the composition of matter of all these molecules. And that's we're the only one who basically hold the patent, and then it's all worldwide from China, Japan, Europe, and Canada and Brazil. So we have all the patent cover on that. We recently also get another patent granted that's based on the work from the DoD, the Department of Defense work. So that's actually set up more further into that patent portfolio. So we'll continue, we're filing more, but we know we're all totally protected. And then the way how we do the protection is we use like a Makushi type kind of claim. So basically it's not one compound, it's like millions compound. It's just anything which touched on the what they call it, generic or something, and then it's got protected. And then we also have this process we call a 220 plus process. This is all proprietary, it's all protected as trade secret. So when we do the project for the smart package for the customers, we don't share any of the process. And we also don't ask any process from customers. So basically, we design molecule, we make the one. The customer usually get is the molecules for them to testing to get the proof of concept. But the process itself, we keep it as a trade secret, unless the customers goes to the GMP productions, the process, the one, and then we will be doing some licensing or some kind of a technology transfer for the customer.
Right, we have a question here. It's when do you expect new drugs soon for breast cancer using your technique?
Well, so it depends on, to really developing the drug, it will be really have to get in the big farms on the side, because they are the one doing the commercialization, the clinic, basically like a pathway. So any drug development would take at least 10 to 20 years. So I would say at least 10 years to 20 years for such drug to get to approve.
And then I would add to that, yeah, CellMosaic is sort of providing the technology for other people to make those advanced drugs for breast cancer.
We do have the breast cancer breakthrough award. That's actually the drug asset we did kind of propose to develop, and that's why the DoD gave us the money. Yes, so we do have part of interest on that part. But we we usually try to collaborate, work with a partner with other people together.
We have another question here about glioblastoma, which I'm unfamiliar.
Yes, so for the brain cancer sense, that is, there are some collaborators which we work, so they do say have some kind of technology which can kind of cross the blood-brain barrier. So it's really depends. Like if you are looking for target specific one, if you have your kind of antibody targeting on something, we can easily help you out just by attach the drugs to that, and then you can try it out. Or if you have some peptide or some other kind of targeting, that can also be done.
Right, the questions are really coming in now. I've got another one here. What inspires you to get up every day and do what you do?
What inspired me? I'm a free spirit person, so I like what I'm doing. So back in the ensemble discoveries, I used to work on a biotech company in Cambridge for 4 years. I'm a very innovative person, so I like to innovate. I like to develop technology. So I had like 6 patents from during my 4 years in that company. But I know that there's not a single one will come out from the commercialization of all this stuff. So at CellMosaic, because the company is my company, I will make sure that the technology can see that, the application side can do that. So this is really drive me, is I saw my customers who use my products, who get a good result, who communicate to me and who thank me about what I have done to helping them out. So it's a lot of driving for that. It's a lot of satisfactions when we're doing that. Especially you can control, how you want to, and in most of companies, it doesn't matter how much patent you have, how many innovation, you will never see the light of them.
I have another question here about post-event sharing of information. And I wanted to make sure everybody knows, yeah, we'll be sharing a full recording of this, and definitely you can send the slides. So that's not a problem at all. Yeah, we'll be sharing.
We can share it, yeah.
Mhm. Okay, we got another question here, but keep them coming. If you have a question, now is the time. Go ahead and add it to the Q&A button down below, and we're here to answer whatever questions you might have. Okay, the next one is about use of funds. So why are you crowdfunding right now?
Okay, that's a great question. So we have so we never actually take too much like fund for so we're my company is organic growth, so we have been for 15 years, so we always have the revenues, and so the technology development majority is funded by either the revenues or the federal fund from DoD and the NIH. As we move forward, right? When now is the time, because we have the AqueaTether, all the process, we kind of build it up. So now we can build up all kinds of AqueaTether linkers we want. So it is really time for us really trying to expand. So we need the fund to buy the raw materials. We want to build up all the AqueaTether drug asset. So which we can show that to the pharmaceutical companies and to other companies. You can try different types of drugs and then to see which one is going to work in your ADC cases. And also in the bioconjugate part, the most of the traditional bioconjugate you see in the market, which is Cell right now, is using all these hydrophobic linkers. So with AqueaTether, we pretty much can replace the majority of them. So basically, we're going to entirely new bioconjugate reagent for the market, including fluorescence dyes, biotins, all this diagnostic reagent. That's the goal. But in order to do that, we do need a fund. So we can't depend on our revenues, because half of the time we're doing the research is trying to support. So I think this is the right time. So why we go into crowdfunding? I think the whole CellMosaic's concept is really supported by the crowd. All my customers is like my crowd, right? So we have so many customers who support us. And then so crowdfunding is really makes sense. It's also kind of improve our imaging on this marketing sense. And we don't want to go to VC. We do talk with VC. But the minute I talk with VC, the first thing they want me to do is, what they want me to do. Just cut everything off. Just do the drug development. Get one asset. Go into the clinical trial. This is not the like the mission I'm set up to do for the CellMosaic. So, and also they're going to take the majority of control, and we will never know this company is going to successful. And then the equity part, they want at least usually 10 million revenues. We're not there yet. We know we're very close, and we just need to kind of explore. That's why crowdfund really makes sense to me. And I also like to work with the crowd and then we got feedback from them, so we know where we're going.
Awesome. I have a few questions are coming, and this is great. One here, what has been your biggest learning opportunity with CellMosaic?
Oh my God. So when you open a company, it's constant learning. So that's the good part is I'm fast learner. I like to learn things. So so I think I started company in 2008. Actually started in 2007. So I said, okay, how am I going to start a company? So what I did is I don't have an MBA, right? So what I do? So I just got all the book from the from the Harvard Medical School, whatever the textbook for the MBA says, from the finance, from the accounting, operation. I just read all the books, and then you set up website. So I just buy. I never know a single thing about the coding. I don't know how to do coding, so I just spent 1 month just read how to do the HTML. So I made my own website, and then I have to write my own write a patent, and I have a really good relationship with my lawyer, the IP lawyer, Phil Zhang. So he used to be our chief IP lawyer in Ensemble. And then he started his own company. So he have me also with the patent side. So we kind of so since lawyer is so expensive, so I have to write the whole patent myself, but he kind of helping me out on the side. So we write like 300 pages of patent, make sure that we covered everything on us, so nobody else can file another patent on this field. So, and then you think that's all done, right? And then now you come up with things about now you have to operate the company, and then you have to raise the money, and then so it's a lot of learning. So it's very exciting. And then the easiest part for us is technology part, of course. That's what I do. That's my bread and butter. The chemical biology, make it bioconjugate, that's all my bread and butter. But there's so whole other things in the business side, which really is a huge learning process. It's great.
Beyond AQT technology, are there other innovative solutions or products in development at CellMosaic? I know you talked about a couple of your other kits.
Right. So AQT technology is the central among all the bioconjugation. So it's not just one compound. It's just a whole system. So we are right now use AqueaTether developing the whole set of fluorescence dye, which can be used for all the diagnostic stuff. So basically you get and then we are also developing the AQT biotin type of system. And then there is other two technology which we just touch a little bit. One is we call OxyLink SS linker, which is for the studying the protein-protein interactions. Basically, that is more for membrane protein, G protein coupled receptor, which is 90% of the drug is basically targeted on the G protein coupled receptor. If you think about how important it for that one is, so that technology is more for the identify target. So it's something in our back end, but it's not right now. Is that it's more like a helping hand. And then we also have another technology called neon technology, which we talk about it. So that is it's totally innovative way to detect. It's opposite for the current industry. They all use a positive eye detection. This one is more for the negative eye detection. So nobody really has that technology yet.
How about reagents?
For the reagents, so this is all the SS link, OX link, and then new. This will be all developing as a reagent and a kit for the customers to use.
Okay. I just have one more question here so far. So if you have any lingering questions, please go ahead and add it now. It's a bit of a pointed question. So it says, how after being in business for 15 years, are you still so small? How many drugs are approved with this technology and how many are in stages 2 and 3?
Okay, so as I said that we are not a drug development company, we are a technology company, the platform company, but we are focused on the product kits and also the partnership. So it's a different business mode. So the technology itself, it's not easy. I would say that it takes years for you for anyone actually to get good technology. To be basically to the point to the one. So, but right now is the time. So right now is the time which we already have all the technologies. So for example, AqueaTether, we started in 2011, starting developing that. And including 2014 DoD Breast Cancer Breakthrough Award, and we'll be continuously developing the process, how to make that linkers to get all the process ready. So when you need these things to do for the drug development, we're not talking about small amount. We need a kilogram run. We need a large scale. So you need to put all these things to be ready. So that takes time. Why so small? So to answer that question, I think you really don't need a lot of people to be innovative. to be, like doing the ones. We try to keep the company small and then focused and developing the technology we needed to be able to expand. So now, as I said, this is the time right now. We're trying to get the money, and then we can do the expanding.
Great. Well, thanks everybody for asking questions here today. I really want to remind you that now is a great time to invest in CellMosaic on Start Engine after you take a good deep dive into learning all about what you're investing in. Yumei, I'm so thankful for you for sharing all of these details about the company, your vision, really how you got started in it. And I'm just so inspired by you as a business owner and scientist. So thanks.
Thank you very much for hosting this.
Absolutely. Check out Start Engine for CellMosaic's investment offering. Yumei, any closing remarks? Why is right now a great time to invest in CellMosaic?
Yeah, I think this is a great time for really invest in CellMosaic because we have all our technologies ready. And we have as I just said, we have our AqueaTethers, all the process nailed down. We're just ready to expand. So if you can come in, this is the best time to, kind of go on the journey to the expansion.
Wonderful. Thanks everyone for attending today. And if you missed it, we'll be sending the recording.
Thank you.